Hi, my name is Kelly Brock, and I'm a student in CS89, Introduction to Deep Learning. Welcome to my final project presentation on variant effect prediction. I wanted to begin with a quick primer about biology. The hereditary information that we inherit from our parents is called DNA, which is your genetic sequence. And in particular, DNA is made up of four possible nucleotides, A, T, G, and C. And in particular, the coding regions of the genome, which are parts that code for what's called mRNA, and that those in turn code for proteins, or as they're called, polypeptides here. This is a region of the genome that's been extensively studied. And in particular, one thing I want to point out is that every single one of us has a bunch of variants that are different compared to most other folks in the whole wide world. And in particular, missense variants occur when your DNA changes in such a way that it actually changes the amino acids that are found in your proteins, which are the building blocks of your cells. Trying to predict the effect of these missense variants, the ones that cause changes in your protein sequences, can actually be a really difficult challenge in the field of clinical genetics. To give you a sense of the scope of this issue, I wanted to point out that roughly 1 in 10 people in the US have a rare disorder, and many of these are genetic. However, many of the missense variants that we've annotated and sent to a database called ClinVar, which is where geneticists can submit variants that they've seen in patients along with their professional determination of whether or not that particular variant is benign or pathogenic, meaning disease causing. Most of the missense variants that have been submitted to this database actually are of uncertain significance. We don't know if they're actually causing a disease or not. Our goal here is to sort of leap into this space and try to use deep learning to provide a computational method of predicting whether missense variants are benign or pathogenic, meaning causing disease. I'm actually going to build 3 separate models, and these 3 different models are going to incorporate different levels of information. The first model is only going to take in what the original, called the wild type amino acid was, and what that particular amino acid is mutating to. The second model takes in a little bit of information about the surrounding protein sequence around where that particular missense variant is located. And finally, for our third model, we're going to actually put in a particular encoding of the full length of the protein sequence, along with information about what that missense variant is. So let's start, first of all, with our model 1. This is the one that's going to take in the minimal amount of information, literally just what the original amino acid is and what you're changing it to. This is going to be a fully connected neural network. And how well does this actually perform without taking into account any of the surrounding protein sequence information at all? Turns out, not so bad. Our training versus validation curves were pretty smooth overall, reaching a plateau around 20 to 30 epochs. And in particular, for our test set, what we're seeing is that our overall predicted accuracy is around a little higher than 70%. When we make a randomized distribution where we're shuffling labels and evaluating based on those shuffled labels to try to get a sense for how well we could do by random chance, we're actually seeing that this test accuracy is definitely an improvement over the equivalent of flipping heads with our particular data. I also wanted to compare against another source of external information. And for this, I used the Blossom 62 matrix, which was first published in 1992. This is a bit of an industry standard. that's very commonly used for aligning proteins from different species together. And in particular, what this is encoding is a penalty, usually used in dynamic programming algorithms, for how bad, in a sense, it is to make a particular missense substitution. More negative numbers means that it is more, it's not as good to make the more negative substitutions, whereas this, it's probably not going to be a huge change. What we're seeing is that we are getting a bit of a correlation, kind of where we expect, where all of our Blossom 62 predicted bad amino acid substitutions are showing up largely as predicted to be pathogenic in our model. If you want more details about this implementation, please read the write-up in my GitHub repo. All right, so we got decent, okay results with this minimal information model. Now, what happens when we start to add in a little bit more information? Now we're going to be putting in information about the missense variant itself, along with a small window flanking it on either side. So we're looking at the surrounding protein sequence around the missense variant of interest. We're actually going to use a bidirectional GRU model to take advantage of the natural language processing capabilities that RNNs have to offer. And what we see is that training it is a little bit noisier and more difficult, but we are potentially getting some improvements in accuracy. And I think that this is a model that could very much be hypertuned even further. Finally, our third model, we wanted to input basically information about the entire protein sequence that this missense variant was found in. The way that I'm going to do that is by using an encoding scheme first published in a 2019 paper by Ali et al. Here. This is essentially going to take in a full-length amino acid sequence, and it's going to compress it down to 64 encodings. We're also going to put in information similar to Model 1 about what the original wild type amino acid was and what we're changing it to. We're also going to put in information about where exactly in the protein sequence that missense variant is located. This model actually had the best performance out of all three models. However, an important caveat is that this good accuracy was actually obtained using a pretty lenient trained test split, where I had to have essentially had the model see that particular sequence encoding before, but not the missense variants of interest. For the previous two models, I was showing a more strict split where you couldn't have seen the sequence before in training. So all that being said, how well does this thing actually do, and what can we do with it? What are the applications? So to that point, I want to present a quick case study now about a gene called KCNQ1. This gene is encoding a potassium transport channel. And the particularly interesting thing about it is that multiple mutations, shown here in this schematic diagram of what the protein looks like, are actually linked to a heart arrhythmia disorder called long QT syndrome. This syndrome can be absolutely asymptomatic over the course of a patient's life. However, it can also cause things like unexpected fainting, and occasionally it can also lead to a potentially fatal heart arrhythmia called torsades de pointes. Therefore, trying to identify patients who have one of these pathogenic mutations in KCNQ1 can actually save lives. However, most of the missense variants that have been submitted to ClinVar, our database that we're training on, are of uncertain significance. So this represents again, a huge challenge for clinical geneticists. All right, so I've already trained my models, and the code for that can be found in the code I'm submitting along with the write-up. There's also a tutorial notebook that should be your entry point into these models called demo all three models.ipy notebook. And I'm just showing a screen cap here because this is the part I want to highlight. If you want to run this notebook yourself, at the very first cell in this particular Jupyter notebook, there's going to be a couple of variables that you'll want to have a look at, and you'll definitely want to change the first one. This is going to be pointing to your directory that you've downloaded the GitHub repo to. So just make sure that you change this to point to wherever your local installation is. As you can see here, I'm actually running these notebooks on Google Colab. So I also have a variable to basically flag whether or not you're running locally versus in this particular cloud service. You can also change the protein that you're looking at to be whatever human canonical protein you desire. You can find the IDs for these proteins called uniprot IDs at this website for KCNQ1. So please feel free to play around and input your particular uniprot IDs of choice. And finally, there's one last thing, which is that if you want to run something besides KCNQ1, the very last section of this notebook is actually comparing to a different external data source that requires manually downloading it. So you want to set this to be false if you're running anything besides our demo protein. So what does our data actually look like? This is going to be for model 1, and we're essentially plotting a heat map of all possible predicted missense variants for the KCNQ1 protein. And red means more pathogenic, blue means more benign. So there's a few interesting things that pop out from here. The first is that we see that alanine substitution, so some amino acid that's going to an alanine, which is this first row here, these actually tend to be pretty benign according to our predictions, and this actually matches something that we expect from biology as well. For model 2, there is another interesting thing about the biology of the protein that we're picking up. KCNQ1 actually has two what are called protein domains, which are sort of their own evolutionarily conserved units. And what we're seeing is that kind of the stripes of red that we're seeing are actually roughly corresponding to those domain boundaries. And in particular, this red stripe is roughly corresponding to the part of the protein that's threading in and out of the membrane. And this does tend to be where a lot of mutations are, so it's reassuring to see that they're all showing up as red in this plot. Finally, for model 3, the predictions on average were a little less confident than for model 2, but overall there was an okay agreement between model 2 and model 3. Again, this model is going to be incorporating information about the full length of the sequence now. And again, we're seeing some mild striping roughly corresponding to those domain boundaries. All right, so if you would like to play around with this notebook, please feel free to do so. The code's available on GitHub. I just wanted to quickly go over some pros and cons. All three models were able to learn something about whether a missense variant was likely to be pathogenic or benign after training on the ClinVar data. Some things that were really important were actually how I split my train, validate, and test sets. And in the write-up, I actually talk about 2 different methods for doing this. Overfitting was also a pretty big challenge. Our ClinVar dataset was actually on the smaller side, about 5,000 labeled variants. So the more data we get, probably the more accurate these models are going to be. And finally, I just wanted to say that I really did gain a much better understanding of deep learning models through this course. And I wanted to say thank you for an absolutely wonderful class.